Hello students, welcome to the academia. Today our topic is immunoglobulin. So obviously it is related with our immune system and globulin is a protein. So it is a glycoprotein which is made in response to an antigen. And this immunoglobulin can recognize and bind to that antigen, which is caused the release of the immunoglobulin. They are gamma globulin in nature and synthesized by the plasma cells. Now, first of all, you should know that what is antigen, what is antibody. So, basically, in our immune system, there are first order defense, second order defense, some local defense responses are there, and some responses are mediated via some proteins. So, we have innate immune system, we have adaptive immunity. Immunoglobulins are basically gamma globulins. So they constitute 25 to 30% of the total serum protein. Generally in our serum, different type of proteins are present. And as we know that the proteins are versatile molecule, a lot of things which are present in our body, either hormones, maximum hormones, then enzymes, carriers, receptors, and in different forms the proteins can present. So immunoglobulins are also some kind of proteins. All antibodies are immunoglobulins, but all immunoglobulins may not be antibodies. So what is antigen? First, the antigen is one substance which is foreign to our body. The substances that enters our body which are foreign, and it include bacteria, viruses, fungi, allergens, venom and other various toxins. Okay, so antigen may be of different types. If we see that what is called antigen, so these are the foreign bodies, the substances which are foreign for us. So the antigens are these, and the antibodies are the proteins produced by the immune system to attack the antigen. Basically, antibodies are immunoglobulins. So previously I have told that they constitute 25 to 30% of the total serum protein. In our serum, different type of proteins are present. Among these, immunoglobulins are one. And in the diagram below, we can see the basic structure, which is composed of 4 polypeptide chains. So in the diagram, we can see the 4 chains, 2 identical light and 2 identical heavy chains, linked by disulfide bonds. Light chains similar in all immunoglobulins. So different classification or types of immunoglobulins are there. In the later discussion I will show you, and they are linked by the disulfide bonds, but the light chains are similar in all immunoglobulins. Light chains occur in 2 varieties, kappa and lambda. Light and heavy chains are subdivided into variable and constant region. So they have some variable region and constant region. Each heavy and light chain contains amino terminal in variable region, carboxy terminal in constant region. So in the diagram we can see in the right side we can see the light chain and also a heavy chain, and both the chains are made up of amino acids. In case of light chain, they are made up of 220 amino acids, and in heavy chain 440 amino acids. So we can easily find out the carboxy terminal end as well as the amino terminal end. Basically, the amino terminal end is known as FAB or paratope. The chains are joined between each other by the disulfide linkages. Now, in this diagram, it is much more zoomed. Heavy chains are structurally and antigenically distinct for the each class. So, we can see the zoom view of the immunoglobulin structure, the heavy chain as well as the light chain. Each immunoglobulin peptide chain has intra-chain disulfide bonds. From the loops, each loop is compactly folded to form a globular structure domain. Light chain contains a single variable domain which is known as VL and single constant domain which is known as CL. So the domains are shown by varied colors. The VL is shown in a bit orangish color and CL in red color. Light chain contains, we can see in the light chain, a single variable domain, one VL and one CL. So in the light chain, one VL and one CL, and in the heavy chain it contains one variable domain and three constant domain. So in the heavy chain we can see CH1, CH2 and CH3, purple in color. We can also find out the hinge region. Hinge region is the segment in heavy chain between CH1 and CH2. So this will be the diagram and all the bonds are SS, disulfide. So these are the, in case of light chain, these are CL and VL, one CL and one VL. And in case of heavy chains, so CH1, VH, CH2 and CH3. So this region is actually antigen binding region. And these are for the biological activity. And both the chains, these are the two light chains, and to have a chains are made up of amino acids. So this is the structure of a very simple structure of a immunoglobulin protein. So in the classification of immunoglobulin based on their structure and antigenic nature. So classification based on structure and antigenic nature of heavy chain. So they are classified in 5 classes. The number 1 is IgG, that is the gamma. Number 2 IgA means alpha. Number 3 IgM means mu. Number 4 that is IgD means delta, and number 5 is IgE means epsilon. So there are 5 classes according to the structure and antigenic nature of the heavy chain. The immunoglobulins are classified. In next step, we see one by one the classes starting from immunoglobulin G. In the immunoglobulin G, there are also some subclasses as well. So in the next video, I will discuss all the 5 classes of immunoglobulin, their structure, detailed structure and functions. So that was the introduction today about immunoglobulin, their types and features. So hope you can understand. Just practice the diagrams of immunoglobulin. If you have any query, question regarding the introductory part, you can ask or can comment in the comment box. In our next video, I will discuss the types in detail.